Hi, my name is Hannah Freed, and I'm a rising sophomore at Princeton University, and for the past 10 weeks I've been part of the Mind Lab as a summer research fellow, working on a computational project to suggest combination therapies for ovarian cancer based on gene expression signatures. As stated in the title, the goal of my project was to suggest drugs to be used in combination with current ovarian cancer treatments based on patient and drug gene expression signatures. As I begin, before I explain how we suggested these drug pairs, I wanted to give a brief overview of the different data sources and applications we used in order to suggest combination drug treatments for ovarian cancer. We used the Mount Sinai Hospital's electronic medical records, gene expression data and survival data from the Cancer Genome Atlas, gene signatures and interactive notebooks from Geo and BioGPs, and drug data and information from Link's L1000 fireworks display. As I stated earlier, our main goal was to suggest drugs to work in combination with current chemotherapeutic drugs. In order to do this, we first needed to define what these common chemotherapeutic drugs are. Using Mount Sinai Hospital's electronic medical records, we searched for common chemotherapy drugs prescribed to patients with a malignant neoplasm of the ovary, as listed in these medical records. From this data, we see that the main drugs prescribed for ovarian cancer have similar mechanisms of action and are traditional chemotherapeutics. Thus, we wanted to find different types of drug treatments in order to make these drugs more effective. As we are basing our drug suggestions on gene expression signatures, we also needed gene expression data for ovarian cancer. We were able to get this data from the Cancer Genome Atlas, or TCGA. Here's a little more information about TCGA for those unfamiliar. TCGA has gene expression and clinical data, such as survival information, for many different tumor types and across many samples. This was created by the National Cancer Institute and the National Human Genome Research Institute. We took the RNA-seq expression data from TCGA, collected from about 350 samples from patients with ovarian cancer, and processed the data to find the up and down differentially expressed genes for each sample. These sets of genes make up each patient's gene signature. We also utilized survival data from TCGA as we were mainly focused on suggesting drugs for the patients with the worst prognosis, which we classified as patients dying within 2 years of diagnosis. Data about a patient's survival was not available for all samples. Only about 2/3 of the samples we studied had information available about their death. The rest of the samples may either still be alive or did not have this information collected. We used the samples from the group having the worst prognosis in the pipeline to suggest combination therapies. To recap, so far I've described the parts of the pipeline for finding common chemotherapeutics and ovarian cancer patients' gene expression signatures. Now I'm going to move to discussing exactly how drugs were suggested. A source that was important to this step was L1000 fireworks display, or L1000 FWD. This web application was created by the Mind Lab using L1000 data that was collected at the Broad Institute for the Links Common Fund NIH program. This web application allowed us to find drug-induced gene expression signatures. This diagram helps explain the most important part of the pipeline and shows just how drug pairs were suggested. At the very left, we see an ovarian cancer patient's gene signature represented by 2 blue circles, and in the center we see the drug-induced gene expression signature found using L1000 FWD for doxorubicin, a chemo drug for ovarian cancer. In order to suggest drugs to be used in combination with doxorubicin, we subtracted the putative effects of each of the commonly used chemotherapeutics. What this means is that, as shown in the diagrams on the right, we removed genes that were assumed to be downregulated by doxorubicin that overlap the sample's upregulated genes. And we also removed genes assumed to be upregulated by doxorubicin that overlap the sample's downregulated genes. From doing this, we created updated gene signatures for each sample for each drug. In other words, these updated gene sets for each sample contain up and down differentially expressed genes that were not already assumed to be reversed by the chemo drug. Then to find drug pair suggestions, we use these updated signatures to query L1000 FWD, looking for drugs that would cause a reversal of their updated gene signature. Our results suggest that chemotherapeutics used in the treatment of leukemia may be useful as combination therapies for worse prognosis ovarian cancer patients. These tables show the suggested drugs to be combined with paclitaxel and cisplatin, which were some of the most common chemo drugs given to patients with ovarian cancer. To find the counts shown in the tables, we subtracted the number of times each of these drugs was suggested for patients with the best prognosis from the number of times each of these drugs was predicted for patients with the worst prognosis, allowing us to more accurately suggest drugs for worse prognosis patients. In these tables, I've highlighted with boxes any chemotherapeutics used for leukemia. Additionally, I also want to mention the top suggested drugs for each of these chemotherapeutics, which are palbociclib and brifaldinib. I will discuss these two drugs more in the next slide. There is evidence to suggest that both palbociclib and brifaldinib, which were the top suggested drugs to be used in combination with paclitaxel and cisplatin respectively, would be strong candidates for combination therapy for ovarian cancer worse prognosis patients. Palbociclib is a drug approved for treating breast cancer and has been shown to increase sensitivity to paclitaxel in ovarian cancer cells. Brifaldinib is a drug that inhibits protein transport to the Golgi apparatus and has been shown to help induce apoptosis in cancer cells. Before I conclude, I want to reiterate that the drugs suggested earlier are just suggestions, and our computational pipeline needs to be refined before experimental validation of our results or the use of our pipeline to suggest drugs for other cancers or diseases. One of the main things we think would be important in refining a pipeline would to be use gene rank from the drug-induced gene expression signatures instead of just using the entire set of genes. I want to take a moment to thank all the members of the Mayan Lab for teaching me so much this summer. Specifically, I'd like to mention Dennis Torrey and Avi Mayan for all of their help and guidance as I worked on this project. Thank you for your time.